The overall goal of this assay is to isolate cancer stem cells from a finite cell line obtained from human osteosarcomas.
The main advantage of this work is to clearly show others in the oncology field how to isolate in vitro cancer stem cells. These cells can then be used to research therapies against osteosarcoma and other solid tumors. Generally, individuals new to this method will struggle if they don't know how to isolate the spherical colonies. Visual demonstration of this is critical, as the isolation step is difficult to realize and to understand. We first had the idea for this technique when we have read about the sphere formation assay described by Gypsy and collaborators. Demonstrating the procedure will be Dr. Gaia Palmini, a PhD student, and Dr. Roberto Zonefrati, a technician from my laboratory.
To begin, prepare all of the necessary culture medium, digestion buffers, and other solutions that will be needed for the procedure. Next, establish an osteosarcoma finite cell line by isolating osteosarcoma cells from a confirmed needle or surgical biopsy, and subculturing the cells obtained as described in the accompanying text protocol. Once the osteosarcoma cell culture has been established and the plate is near confluence, remove the medium by aspiration and trypsinize the cells to release them from the plate.